Okay, hello English speaker colleagues. I'm here today to tell you how you discover a variant of SARS-CoV-2 virus, and you can do this easily by using Biopython. Okay, so Biopython is a tool that is, well, a set of tools that help us a lot with bioinformatics. So here I just will place you some uh, biological background, very superficial one, just for you to understand how or what I'm doing there, right, during the discovering process. So there are two websites you should visit. One is NCBI, and the other one is BVBRC. So these ones will show you the variants of concern, of interest, and you will get samples, anonymous samples from any variants you would like to test or observe or do any kind of study you would like. So I'm going fast here just for you to get this background. So let's go. So what you're seeing here is the Wuhan, China original virus genome. So you see 29,000, almost 30,000 nucleotides involved. So this is the original virus. And why I'm showing the original virus is because if you want to, that's because you need to have a basis to compare the variants to. So that's the point. So I would just zoom this in to show you a little bit else. I believe you can see a lot of letters, H, G, C, T, and so on. So these are the genome's normal sequences, nucleotides, right? So you can apply all the rules you know from genetics, from genomics over here. So this is a complete genome from the original virus. And all the vaccines are made on top of this one, okay? Then later, I will explain to you why Omicron was considered a concern to many scientists and the vaccine. But it's fortunately, covering these variants as well. Vaccines are covering them. Don't be afraid of this, technical terminology here. I won't be explaining that in details. What I can, take away from this slide is that the coronavirus 2, because SARS-CoV-2 is one of the families of coronaviruses, so it's not a single virus. This one has 11 genes. If I'm not wrong, the SARS-CoV-1 had 6 or 4. So this one is kind of more complex, let's say. And in yellow, well, here you have all the sources, the technical sources, if you want to go through and get deeper. But the numbers you see is the position of that beautiful string I showed you before with 30,000 nucleotides. So the position, let's say, the spike protein, which is the one that we are being focusing on. And why? Because the vaccines are made on top of this one. So you take the genome substring from these positions here, and then you compare the variant or if that is the original Wuhan virus. So if you count all these yellow genes, you will count 11. That's what I'm saying, right? So from now on, you see, you have nucleocapsid proteins. This is also present in other viruses and etc. Membrane protein and so on. Here, the fastest response from the laboratories and from the universities and from the scientists mainly was on top of spike, because spike is the protein that coronavirus uses to enter your cell and gets reproduced. It depends on a cell. It's not considered a full living organism, so it depends on a cell to get reproduced. So once it gets reproduced, it causes all that, horrible things about the respiratory and also, maybe let's see, neurological stuff, neurological issues. I mean, wherever this virus comes in, since it's a virus that is new to the human being, it will cause problems. That's the truth. right? So here we go. Let's continue here. So what I'm doing here is the spike proteins can be generated from those substring positions there. I won't go very deeper here, right? But there is a dogma from biology, the dogma from the genomics we apply here, which is DNA we can transcript to RNA or vice versa, and then from RNA we can translate them to proteins. So that's why you see these other letters here, which are not A, C, G, and T only. So just to remind you, in case you already studied or if in case it's new to you, so if you go to the transcription, if you go from DNA to RNA, whatever T nucleotide is transformed into a U nucleotide. And then the combination of three of them composes a protein. And then when you have let's say a chain of those proteins, you have what we call peptide, right? So the peptide is a group or a set of sequential proteins that truly identify as a virus. So every virus has this mapping, and that's why people sequence genomes. That's why they sequence DNAs. Got it? Okay. I'm just citing a source from Dr. Werner Gitt, just to tell you that DNA is not aleatory or random combination that I showed you, the 30,000 genome big string. It's actually a very, very complex language, right? It's more complex than the language we talk or a programming language, because it's considered apophetics, because if you change one gene, you can change a lot of proteins, and the response will be another one, and very precise one. So this is the top one in complexity of information, according to Dr. Werner Gitt's book. There are people who disagree with that, but I just place it you as a curiosity, right? So here we are. I won't explain codons, those things. You can go through the dictionary and or a biological website to get you in touch with them. But what really matters to us is that we have vaccines based on spike protein. Okay, and here we have the original Pango lineage. Pango is from pangolin, which is believed to be the animal where the virus jumped into the humans, firstly, in Wuhan, in China. And then many variants came in. Why do we have variants? Let's understand this. Variants are there because whenever we have an environment with a virus that is not treated or stopped or eradicated by a vaccine, this virus surely will try to find chances to escape the vaccine and become more powerful. Yeah, you may call this intelligence, you may call this anything, you may call this random thing, but actually the viruses do that, and they try to be more powerful. They try to escape, vaccines, or escape any immunological or immunity you may have, and they will try to reproduce themselves by using your cells. And once they get reproduced, they cause all the deaths, all those things that we are tired to hear, unfortunately, through the news. That's why it's very important to vaccinate. If you don't do that, what happens is that you get the virus more chances to get more powerful. And Omicron was a concern. I will show you later during the programming. Omicron has a lot of changes when compared to Wuhan proteins and spike protein. And there was a concern about whether the vaccine would cover that or not, right? So what is the only way to get this virus eradicated? By vaccination, just like you do with measles, with smallpox, with other viruses as well. So whenever you narrow down the window for the virus to try to evolve itself, the best you will be. So once one person gets vaccinated, the person is helping others around. And the opposite is very true. Vice versa. If many people don't vaccinate, they are the best field for the virus to find someone who is not vaccinated and then reproduces and then gets more powerful and then generate more variants. So that's why if someone gets vaccinated, the person is helping out the person himself or herself and also the community around. So if someone is not vaccinating because I don't want this vaccine, it's experimental. First, it's not experimental. It's official. It was really tested. There are lots of technical papers showing this. So no one is putting vaccines here that will put you in danger, right? They are very secure. And if the person doesn't get vaccinated, this will be the focus for the virus to get more powerful and get reproduced because the virus wants to persist. It's important for you to understand that, okay? Okay, so let me go ahead. So this is just a literature that I would like to indicate to you. It is written on a leisure language, just like I'm saying here. I'm not going so deeper. And this book was very great because it helps you stop thinking on conspiracy theories first. And secondly, it shows why we are in danger with this virus and new virus that may come and what kinds of measures we can take. And it shows also the mechanics and the story behind a virus evolution and a virus evolution and a virus, let's say, variant surge. Okay, so here we go. I will just show you the first demo, which is the Python. I have a code in Python with Biopython, which is very efficient to test the proteins and tests from comparing the test I have with the Wuhan. As I said, you need to compare to the original one. Here we go. So let me start up Jupyter Notebook, which is my Python environment, and I have Biopython on top of that. So here we go. So I will open up, it will open up my browser here. And here I am. I will go to my COVID-19 directory and I will go to my program. And here we are. So here, before we go, there are some things I'd like to show you that are very important, and you can do it from home. You don't need to be a specialist on that. If you go to this one, this link, this is the NCBI virus. This is the GenBank, which is very, very rich. Here you can find many. Here I placed Brazil, which is where I live, but I can't take off Brazil from here. And there are many proteins. Do you know the proteins that see? The proteins here. And I will go to surface protein, which is the spike protein, because my program was made on top of spike protein because the vaccines really go through that. Is it possible to build vaccines on top of other vaccines? Yeah. What they need to test is the efficiency if considering other proteins. That's why we have so many testings before placing a vaccine available to the public in general, right? So here we have, samples from everywhere. Japan, USA, China, you see above in the top of the list, and here we go. And there are lots of them here, and you can choose, right? So one thing you can do, and one question you may have right now, let me go to the beginning, is how my PCR test, which is a genetical material, is turned into a file like this. Let me go to the FASTA file, which is a sequence of a protein. That's the same as I take that part of the genome corresponding to the spike protein, transcript to RNA, and then translate into proteins, and then I get this, right, as you've seen. So how can I go to this from genetical material into this? So go to a website called illumina.com with double L, illumina.com. There are many, many devices where you place the material genetical things with some reagent, chemical products, and also some photon, insiding on top of that. And you will see those material genetic being transformed into a FASTA file. So the output of that, the person can connect it to the internet or place a micro SD card or whatever, and the person will get a plain text file with this combination here, with this information here, not a combination. And then, of course, you can, if you want to download it, you can download it. And I choose FASTA because but Biopython is prepared for that. They have libraries to that. They have libraries for other things here as well, but you can download it and test to discover the variant. We don't know the variant here. What we know from this information is that the person is really infected with coronavirus SARS-CoV-2, right? The second website here is just an informational one. It will show you, it's the bv.org. You can rewind this video and go there. But the bvrc.org will show you the variants of concern. There are two classes of variants. They are the variants of interesting point, and we have variants of concern. Variants of concern, you see this list is going, bigger and bigger each day until we have a massive vaccination around the world. The variants of concern are the ones that scientists look deeper into, that they really put a spotlight on to really know whether the variant may escape the vaccine or not. We are lucky because Omicron doesn't escape the vaccine. Now scientists are working on maybe kind of spray or medicine or other vaccines that can inhibit the transmission, because the transmission is not inhibited yet. Yeah. What happens is that if we are vaccinated with all the doses, we won't go to an ICU, or the person that goes to an ICU, no vaccines protects 100%, but they protect a lot of it, around 90s or 80s, which is very big. Even 50s are considered very big, good from WHO, so the World Health Organization. And here, what you see, if you want to know the namings and what is changed across the proteins, I placed this in my program. I'll go there now and show you. So let me go there. And here I have 6 variants I placed. And what I need to compare, I need to compare the Wuhan protein sequence, the original virus one, with any testing and trying determining the variant that is there. That's the point. Okay. So here I place exactly that data from this website. So if you see age 69 and the minus sign, it means to me that the original age protein was deleted by the variant. So nothing is placing that in the position 69 for that protein in the FASTA file, right? Likewise, what we have here is P in the position 26 being replaced by an S, right? And so on. So that means to me that where I need to compare. So I will have a percentage, and the biggest percentage will tell me which variant we are talking to. Sometimes it's not classified. You see a lot of unclassified, or you see a predominance of a variant. Okay, so let's test some ones here. So what I did is I created, a dictionary always containing the Wuhan spike protein and the changes for each of the variants, right? And I will show you 2 things, very important things here. That first, variants are, I will give you the spoiler right now. First thing, variants are circulating worldwide, right? And secondly, Omicron has much more changes on the spike protein than other variants that was ever identified. So let me go ahead. Here I have a file. I placed one from Brazil. Let's run it. Let's see what happens. So I created the functions that detect the variants. So what the program is going to do is it will read the two dictionaries from Wuhan and from the testing that I downloaded from NCBI. And here we go. From this one, the UK variant, I have 55% of matching, right? And you see, pay attention to that, we have 11 mutations comparing to the Wuhan proteins from the spike protein. Okay. Oh, I got 100%. So that's the Manaus Brazil P1. Okay. That means, oh, all of this is from 2022, this year, right? That means that this variant is still present. So we need to get vaccinated. That one was the one that caused a lot of problems in Brazil during 2021, which was known worldwide. Is still here, right? Indian Delta variant, so it's just 17% of chance. South Africa, first South Africa variant from last year, you see 58%. So that means to me that it has some correlation with this one. But surely if you get 100% on a biological thing, that is that variant. You don't need to have any doubts on that, right? There is a California one, which was a variant of concern, was not so well known because it stayed across California. But yeah, it's so you see just 4 mutations here and see the Omicron there, which is nuts, right? You see 39 changes. That's why scientists got so crazy, because they didn't get crazy. They got concerned because with such a number of mutations here, it's, you know, very dangerous that one variant may escape the vaccine, right? Fortunately, this is what didn't happen. Right? If you are vaccinated, if you are not vaccinated, any of these variants may be lethal to you. And we know many cases of people that didn't have comorbidity problems and anything, and many people died. And I lost relatives on that. As you may know, or you may have relatives you have lost on this. So don't play with this virus, right? Don't kid this virus, because it's very dangerous, right? And it's not political, you know, it's a scientific thing. So okay, we got Brazil one. Let's pick up one and USA one. Let's see what happens. I picked up this randomly from NCBI. Let me run again and see the testings. And here we go. Let's see what I have. It's running. Oh, this number here means that the proteins have 1,000 to 700 big number of strings in my protein sequence there, right? So it's 1270 big, right? So here we have a percentage that it may be the UK variant, and then we have 0, it's not Brazil for sure. We have a small percentage for Delta variant and so on. We have some percentage for Africa, and we have 0%, and see the Omicron here, it's 100%, so it's the Omicron one, or Omicron, I don't know how you pronounce this in English, but it's the new one. So you see, if you go to NCBI, you see lots of samples on Omicron. You will find them on NCBI as BA.1 or BA.2. That's the naming they place it on GenBank, right? Let's place another one for you to see it. Okay, last one. I got one from Romania, if I'm not wrong. Oh, here are the functions. Let me go a little bit down. And here we go. Demo Romania. Just pick it up that randomly and try to see what happens. Here we go. I'm running that again. And it reads very fast. It reads that very fast. So we have one, 1273 in length for the proteins. We have 36% of UK. You have 8% for Brazil. May have some or one or another gene that is matches Brazil variance changing, right? Oh, but it gets 100% on the Delta variant. So this is surely the Delta variant. Let's see the others. 33% for Africa, 50% for California, and 21% for Omicron, right? So if you get 100% in biology, it's not an exact science like computation, like mathematics, but it's surely you can indicate it's a Delta variant. And in fact, when I picked it up from there, electorally, randomly, it was written in front of in the other column in front of there was telling that it was the India variant. That's it. That's it, guys. That's it, people. That is how you can use Biopython to detect variants of coronavirus. Thanks for watching, and I hope this information is useful, and feel free to use that to convince people who don't want to get vaccinated the kind of danger they are inside, and they don't have the idea on the size of the problem they have. Okay? Thank you so much. See you.